Cellular respiration is a series of reactions that takes place in the mitochondria of a cell. It uses nutrients to convert, well, it uses nutrients like glucose and oxygen to make energy with CO2 and H2O as byproducts. The series of reactions are glycolysis, which turns glucose into pyruvate, and which is an anaerobic reaction. And the next steps in the process are pyruvate oxidation and citric acid cycle, which are both aerobic. And in the end, and the final part of the reaction is the oxidative phosphorylation, which is also aerobic.
Glycolysis is a series of reactions that breaks down glucose into pyruvate. So it starts off with a molecule of glucose, which gains a phosphate group from ATP, making it chemically reactive and fructose 6-phosphate, which then gains another phosphate group, making fructose 1,6-biphosphate, which then forms dihydroxyacetone and glyceraldehyde. And this reaction is continuous and it never reaches equilibrium. And G3P, which is glyceraldehyde, is used in the next step, which it is oxidized by transfer of electrons to NADH, forming NADH and two hydrogens, forming 1,3-biphosphoglycerate, which then gains a phosphate group from ATP, forming 3-phosphoglycerate, and then which forms 2-phosphoglycerate, and then two water molecules are released, forming phosphoenol pyruvate, and then it gains 2 phosphate groups for each pyruvate, forming a pyruvate molecule, 2 pyruvate molecules.
So overall, glucose, for each molecule of glucose, 2 ATP is used to form 4 ATP. The glucose is broken down and becomes 2 molecules of pyruvate and 2 water molecules. 2 NAD molecules and 4 electrons, as well as 4 hydrogen ions, are transformed into 2 NADH molecules, and 2 hydrogen molecules are left over.
Okay, the citric acid cycle is a central driver of cellular respiration. It produces 1 ATP per cycle, 2 ATP per glucose, 6 NADH, and 2 FADH2. In step 1, the 2-carbon acetyl group of acetyl CoA is added to the 4-carbon oxaloacetate to form the 6-carbon citrate. Step 2, the citrate loses a molecule of water and another is added. In the process, citric acid is converted to its isomer isocitrate. In step 3, isocitrate loses a molecule of carbon dioxide and is oxidized, forming the 5-carbon alpha ketoglutarate. In step 4, alpha ketoglutarate is converted to the 4-carbon succinyl CoA. Step 5, CoA is removed from the succinyl CoA molecule and is replaced by a phosphate group. The phosphate group is then removed and attached to guanosine diphosphate, thereby forming guanosine triphosphate. The final product from the removal of CoA from succinyl CoA is succinate. In step 6, succinate is oxidized and fumarate is formed. Flavin adenine dinucleotide is reduced and forms FADH2 in the process. Step 7, a water molecule is added and bonds between the carbons in fumarate and are rearranged, forming malate. In step 8, malate is oxidized, forming oxaloacetate, the beginning substrate in the cycle. NAD plus is reduced to NADH and H plus in the process.
So overall, the citric acid cycle will generate 2 AT pure glucose, 6 NADH molecules, and 2 FADH2 molecules.
All right, so here's the electron transport chain. This is where almost all the ATP that's produced in the respiration is where it's produced. Almost 26 to 28 ATP here is produced. So, and it's created through oxidative phosphorylation. Now, let's begin with the NADH and FADH right here. Now, these are produced from glycolysis in the citric acid cycle, and they're very important. So, they give up their hydrogens right here, and they combine with the protein complex to give up their hydrogens, and then they turn to NAD and FAD respectively. Now, these hydrogens go up here, and we'll use them for later, but also they give off an electron carried through these electron carriers, this represented by the yellow arrow, and it goes all the way to here, which combines with 2 hydrogen, which we got from glycolysis, and oxygen, which we got from the cell. And that forms water, which is an important product. And also through this reaction, we also get an extra hydrogen, which goes up top. Now, the energy is required to move this hydrogen up here, which is provided by these molecules. But all of these hydrogens move here into the process of chemiosmosis. Now, in this protein, the hydrogens are moved through and they generate energy, almost like a turbine, through this protein called ATP synthase. So they move through this protein and they create energy like a turbine. And through this energy, that uses the energy to combine the ADP and the individual phosphate group, which combines to make ATP. And that is how all of the ATP in the chain is made.
We already did the summary. We're done.
Yeah.